All right, so without further ado, let me introduce what we're doing today. This is our speakers, Laura Hercher and Carl Zimmer, who will be presenting Hope, Hype, and Horror Movies Contemplating Human Germline Modification. Laura has taught courses in ethical, legal, and social issues in genetics for more than a decade at Sarah Lawrence, Joan H. Marks Program in Human Genetics, where she is the Director of Student Research. She served as chair of the National Society of Genetic Counselors Ethics Advisory Group from 2010 through 2013, and is a co-author of the NSGC white papers on stem cells and prenatal testing for adult onset diseases, as well as numerous statements, including the NSGC position on somatic cell nuclear transfer, that is, cloning, incidental findings, and testing minors. Laura is a founding member of the frequent contributor to the DNA Exchange, a blog for genetic counseling community. She has published original research, commentaries, and science journalism, including articles in Scientific America and Wired. Her first novel, Anybody's Miracle, was published in 2013. Carl Zimmer is a columnist for the New York Times. He is the author of a dozen books, including Parasite Rex and Evolution, Making Sense of Life. Carl's writes regularly for magazines such as National Geographic and The Atlantic, and is the national correspondent for STAT, a new publication about biomedicine. He is a three-time winner of the American Association for Advancements of Science and is the 2015 winner of the National Association of Biology Teacher Distinguished Service Award. If you wouldn't mind, and please welcoming them to the stage.
Thank you very much. Good morning. We're going to start actually by polling you guys and just to find out sort of your views on the issues that we're going to dive into today. In other words, germline engineering. Laura and I are going to be taking you through some of the specifics about what's been happening recently, and but we want to get a kind of a sense of the crowd first, and then at the end we're going to ask you the same questions, and you're welcome to change your mind or not. So this is the question we have. Which forms of human germline modification do you believe should be permitted? So A is none. Human germline modification is unethical and should never be allowed. B is experiments on early embryos where there's no reproductive intent and the embryos are not allowed to develop past the blastocyst stage, about 14 days. Or C, in the future, carefully controlled and monitored experiments involving human germline modification may be worth doing in some circumstances. So we're just talking about experiments. Are we talking about like reproductive use? Okay. So so C is you want to design babies, you can design babies. So how long do we let people? I would go ahead. Okay. Well, so far designer babies are winning by a landslide, which is fascinating. I guess you're a pretty forward-thinking crowd, maybe? I don't know. And we have another one? Yeah, there's one more. There's one more, okay. Now this one, so let's switch to the next one. Now we're going to talk about mitochondrial replacement therapy, and Laura's going to talk about that in more detail, and that's essentially where you're treating mitochondrial diseases by basically putting in healthy mitochondria, which have their own DNA, into an egg. And this is, the subject of a debate right now, and it's been considered in Britain. I guess it's been approved, but Laura will just fill us all in. Anyway, A, mitochondrial replacement therapy. Well, I should back up. This is when people want to sort of be trolling the issue, they like to call this three-parent babies. So how do you feel about three-parent babies? Not to jinx you on it. Mitochondrial replacement therapy is germline modification and should not be permitted. That's A. B is mitochondrial replacement therapy is not really germline modification and should be permitted, assuming appropriate preclinical testing suggests it's not too risky. C, mitochondrial replacement therapy is human germline modification, but that is no reason it should not be permitted, assuming appropriate preclinical testing suggests that it's not too risky. So we're sort of asking you about whether you approve of these things and also kind of how you view these. This kind of stuff gets philosophical pretty quickly. And as I've been talking, again, C is way ahead. Should I wait any longer? Let's move on. Okay, well, thank you for that data. So, when we talk about these sorts of issues, it can be a little hard to sort of feel like that we're really talking about something real or something potentially real. It sounds like science fiction. But the kinds of things that you've been talking about, at this meeting, for example, like, oh, let's just grab DNA out of a mother's blood and figure what the baby's genes are. That sounded like science fiction a few years ago, I'm sure to you. It certainly did to me when I was reporting on the earliest stages of that research. So the strange thing in this field is that science fiction becomes science, becomes medicine very quickly. And I'm going to talk about a particular example of that in the time that I have. This is, like I said, this is not a movie, and to give you an example of why this is really actually a very serious issue, we can just talk about the case of Tyler Zielinski. Tyler died in 2012 at the age of 20. He lives in upstate New York, and he had Duchenne muscular dystrophy. He came to some fame because a lot of celebrities would, interact with him and sort of draw attention to his case. He fought really hard against this disease, which affects the muscles. He was able to graduate from high school, but then the disease claimed him. And this is a disease which is horrible, but conceptually it's very simple. It affects dystrophin, which is involved in muscles, and basically all that happens is that there's an effect on one gene. And this is just one example of a muscular dystrophy mutation. Basically, it knocks out a portion of the gene so that it cannot properly make the dystrophin protein. That's it. That's all. So imagine, if you would, that when he was still an embryo, if you could have gone in and just switched that faulty gene with a working version, you would be a totally normal kid. Other than that, in other words, that's all it would take to have given him a full life. Now, and like it's a deep question. Like, would that be okay to go in and change that? It is possible now to talk about this seriously thanks to a new technology called CRISPR. And CRISPR, among other things, has the distinction of being the method with the worst acronym ever invented. And I'm trying to talk to you about the future of the human race, and you're thinking about some sort of candy bar. I don't know why they decided this would be a great name, but this is what it actually stands for: clustered, regularly interspaced, short palindromic repeats. There's a very long, complicated story about how that got to be that way that I'm not going to get into today. I wrote an article that came out earlier this year in Quanta, which is published by the Simons Foundation, where I kind of went into the deep history of CRISPR. CRISPR is actually a defense system that bacteria use to kill viruses. And amazingly, we can use it to edit our own DNA. Nature is weird that way. If you would prefer to listen to the story of CRISPR as opposed to read about it, I went on Radiolab not long ago, and I talked with Jad Abumrad and Robert Krolwich about this, and we kind of went back through the whole story. So the episode's called Antibodies Part 1. So basically, what matters to us today is that once Jennifer Doudna at Berkeley, Emmanuel Charpentier at Max Planck were able to figure out how bacteria use CRISPR, they and other people like George Church at Harvard and Feng Zhang at MIT said, hey, wait a minute. These bacteria are targeting specific sequences of DNA with great precision. We could do that too. We could take this stuff and manipulate it as a tool. And this is a video just to sort of kind of let you visualize what I'm talking about. So basically, it's very simple. There are two pieces to CRISPR. There's a big enzyme that's going to make a cut at a particular place in DNA, and it's actually cradling a piece of RNA that's single-stranded, sort of counterpart to DNA. And that RNA has been engineered to match that site in DNA that you want to cut. Okay? So here in this video from the McGovern Institute, where Feng Zhang is, it's showing you how that RNA is sort of cradled in there in the enzyme. And so you deliver it into a cell's nucleus, and these enzymes are bouncing around, and they will only lock into one place, that one site that you want to cut. And basically what happens is it pulls apart the DNA. It essentially sort of compares it, makes sure it's in the right place. I'm in the right place. I'm going to make the cut, slice, and then it'll cut in another place, and you've chopped out a piece of DNA. So let's say it's that bad dystrophin gene, gone. Now, if you just cut the DNA, it's going to repair itself, and it's going to heal itself. Now, in that case, you're just knocking out the gene. Now, you can just add at the same time some DNA that's the correct sequence. If you do that at the same time, then it's going to basically seek out this cut section of DNA, and it's going to fall into place. And then the DNA itself is going to build up the second strand just on its own, thanks chemistry. And now you've got a working version of the gene. I just explained it to you in I guess, about a minute. It's really not that complicated. And just to give you a proof of concept, we can talk about Duchenne's muscular dystrophy, this particular case that I just introduced you to. So just last year, Eric Olson and other researchers, what they did is they created a mouse model of Duchenne's muscular dystrophy. They, in other words, these mice have that problem, that defect in the gene. And while they were still embryos, they used CRISPR to insert a working version of the gene. Now, these embryos became mosaics. We had a fantastic session yesterday about mosaicism. They were mosaics. In other words, some of the cells had the working version of the gene and some didn't. That was enough so that when the mice developed, they were healthy. In other words, they cured Duchenne's muscular dystrophy, at least in a mouse. So, like I say, this isn't science fiction. This is working, at least in animal models. And there is more work that is in the process of going forward to where scientists actually want to do research on human embryos. And so actually at the Francis Crick Institute just last month, there was a request made to the United Kingdom government asking for permission to edit genes in embryos, human embryos, specifically just to look at how genes help with fertility. Not to create designer babies, but just to understand development better. That decision's pending. Now, this stuff has been happening so fast that a bunch of scientists, including some of the people I just mentioned, like Jennifer Doudna, came out in April with a sort of position paper about research on CRISPR and particularly on it in the germline. And they said, whoa, we should just have a moratorium for now. We should not deal with this. Little did they know that in China, some people weren't paying attention to their concerns and had actually already done it. And so this line has already been crossed. This was a paper that came out in Protein Cell right around the time that that statement paper came out. What's interesting, I talked to Jennifer Doudna what she thought about this paper, and she pointed out that there were problems with it. The results were not great. They tried to fix a gene in human embryos, and it sort of, to her, it kind of makes her case. In other words, in a lot of cases, it didn't actually edit the gene they wanted to edit. It put the gene in the wrong place sometimes. You don't want that, because it might get regulated in a way you don't want. Then another issue, which is not germane to this particular study, but which is a general concern, is like if you start tinkering around and just putting in genes that you think are good, they might be good for one function, but they might have harmful side effects, even if you get the CRISPR procedure just right. So, that's fair enough that this particular experiment wasn't great, but it was a very early, very simple version of CRISPR. And CRISPR, in that time since they did this experiment, is shot forward. CRISPR is getting better, and it's getting better fast. So just to give you one example, I just wrote, I guess 2 weeks ago, about George Church's most recent project, where he and his colleagues were engineering pig cells in the hopes of being able to make pig organs that would be suitable for human transplantation. And one of the concerns is that they have these sort of virus DNA sequences embedded in them, and you don't want pig viruses suddenly coming out and making you sick once you get a liver from a pig. So they used CRISPR to go in and inactivate all the viral DNA sequences, 62 of them. So basically this is doing 62 different CRISPRs at once. Now, a caveat is those were 62 of basically the same genes, because it's the same virus with many copies. But he's got another paper in the works, apparently, where he and others have actually engineered 20 different pig genes in the hopes of sort of dealing with rejection. So this just shows you that the concerns about the risks of CRISPR, I think, are kind of, in a sense, kicking the can down the road. What are we going to do when CRISPR does become safe? What then? Because that's when the real challenges begin, I think. So in talking with actually some genetic counselors at this meeting, I've been saying, like, well, I came in here thinking, like, is this really going to be really necessary? Is this just going to be just a few cases where you could really justify doing this? Like, hey, pre-implantation diagnosis, that'll take care of everything. You just pick out the embryos that are fine and you go on your way. And I really appreciate that experts here have actually informed me that it's not so simple, that you don't just magically get 1 million embryos and you can pick out the very, very best ones. Sometimes you don't get any that are the best. That might be a situation where parents would say, well, couldn't you just fix that one? And the answer will be yes in a few years. I mean, if we decide that that's okay. Technologically, the answer will be yes. But then we get into, if that happens, then we're going to get into trickier areas. And I'm just going to close my comments by talking about a couple tricky areas. So, okay. So your doctor's like, okay, we're going to go in there, we're going to fix that dystrophin gene, because you don't want that. But tell you what, while we're in there, how about we give you an extra fix in there? Sort of like you buy a car and you want air conditioning, and then you want the Bluetooth. You're like, what do you want? So, for example, would you, if you had it in your control, would you prevent your child from developing Alzheimer's? Millions of people develop Alzheimer's. Just here's 2 examples: Rosa Parks and Ronald Reagan, who both developed it. Now, there is a rare variant of a gene, APP. We know about this gene. It's been demonstrated that it is extremely protective against Alzheimer's. And people who get this protective allele, they're totally healthy otherwise. So what's the harm? Like, your kid's not gonna get Alzheimer's. That'll let you sleep at night, right? I mean, it's not like you would, if you knew that your kid was gonna get Alzheimer's, you'd feel very happy about that. And then the last example I want to give you has to do with a gene called Klotho, which has been shown to boost cognition in humans. It basically slows the decline in cognition as you get older. And in studies on mice, it actually doubles their performance on certain memory tasks. So, okay, so we've made sure your child's not going to develop muscular dystrophy. Kids are going to be protected from Alzheimer's. Just, for an extra $200, we're going to throw in extra intelligence. I mean, because, hey, you're just going to send your kid to, a tutor or something when she's in high school, right? Let's start this early, shall we? And in fact, I predict that we might be in a situation where if we all decide this is okay later on, I mean, maybe this will become sort of like vaccination. Okay, so vaccination started out as experimental, but now it's like, I think that certainly I vaccinate my children. I hope everybody else vaccinates their children. It gives your kid a better start on life. And so, maybe people are going to be sort of feeling like, well, why wouldn't I do this? What kind of a parent would I be if I didn't give my kid all the advantages in life that I could? These are tricky questions. And so I just want to ask you a couple more poll questions now that's the end of my talk. Okay, so there are going to be these 3 examples I gave you. So, number 1, is it acceptable to use CRISPR for germline engineering to cure muscular dystrophy? So this is the Tyler example that I gave you. All right, and the no's are starting to creep up. Okay, so I'm talking, and think about this, like, you're gonna, like, the kid who is, cured or engineered to not have muscular dystrophy will pass that gene down to his or her children. You're affecting heredity. You're making a permanent mark on our gene pool for generations to come. And that seems to not bother you at all. Okay, look, all right. I'm calling this one, you have voted in favor of yes overwhelmingly. There's no chance that the no's are going to catch up at this point. So let's go to the B, please. Is it acceptable to introduce a protective allele for Alzheimer's? What I was just telling you about. Whoa, okay. So now things are different. And maybe in the questions answer we can talk about why is that different? Again, people already have this gene. Only in Iceland for some reason, but they've got it, and they're fine. They're happy Icelanders, okay? You're not going to be harming your child by giving them this gene. You're not. And the chances are your kid will be less likely. But it's looking like no, not quite as overwhelmingly as yes before. Interesting. Let's move on. Okay, last one from me. Clotho. You want to give your kid Clotho. It's Clotho or SAT prep. Come on. The point is, like, we if you say, like, oh, I don't believe in tampering with the biology of my child's mind, it's like, sending your kid to school changes your child's mind. It's like you are rewiring your child that way. I'm not endorsing either way. I just encourage you to sort of think about the real sort of ramifications of what is happening in these different cases. But no. Well, yes, a little, but no, not really. And the yeses are really losing out a lot more than before. Okay, so that's it from me. I have a little thank you slide, but I'll be happy to help answer questions afterwards.
IVF may be cheaper than SAT prep, I don't know. Hi, good morning. So I'm going to talk a little bit about going back, some of the concerns we have about human germline modification and where those concerns come from and so on. Concern over the potential harms of altering our DNA is not new. Anxiety accompanied the very first experiments in recombinant DNA therapies or just recombinant DNA work at all, and led to a moratorium on all work worldwide leading up to this famous conference in Asilomar in 1975, when the entire field agreed on self-imposed regulations to limit risk. Asilomar is often held up as a model for today, but it should be noted that everyone in the field at that point in time could fit around a conference table to talk, and that pretty much they could all conduct that conversation in English. That community today includes many more people from many more countries, from many more institutions and companies, and possibly some basements and garages. But don't worry, I hear they're only using it to make beer. Concerns specific to DNA modification crystallized a decade later as a part of the debates over gene therapy. In 1985, the NIH Recombinant DNA Advisory Committee, the RAC, published guidance that made a distinction between gene therapy that changed somatic cells, which was fine if approved, and germline therapy, which was not. This was guidance, not law, not a regulation, a restriction on what could be paid for using tax dollars, but it drew a line in the sand, and it said that that line is the human germline. That's the line we won't cross. That limit separated gene therapy from some of the scarier aspects of what Time magazine labeled the Biotech Century. It said, we can do this without opening the door to designer babies, without ensuring, as social critic Jerome Rivkin said, our way of life is more likely to be fundamentally transformed in the next several decades than in the previous 1,000 years. Sorry, I said that like, to be more fundamentally transformed, yeah. And for 30 years, we have lived with germline modification as our line in the sand. What exactly are we afraid of? Change is scary. Every generation is frightened that the world they hand down to their children will be irrevocably changed. And every generation it is. Beyond our inchoate, if understandable, fear of change, what are the specific dangers of taking it upon ourselves to alter the DNA of future generations? This is what Time magazine called biology's splitting the atom moment. Can we try to define those fears to quantify the risk? That's a tall order, but let me try. Well, I like to divide the risks into two categories: the Frankenstein issues, or what happens if it goes wrong, and the Gattaca issues, or what happens if it goes right. For all my students out there, I'm going to just like out myself right here. I've never seen Gattaca, okay? I just talk about it all the time. This is true. All right, the Frankenstein issues. Well, what are the things that can happen if it goes wrong? And Carl touched on some of these. Mosaicism, converting the DNA in some cells and not others could create a mosaic embryo, making confirmatory testing difficult, as you guys know, and creating a risk for future generations. Off-target effects, different types. One type, the DNA is altered in more places than you intended. You meant to do one thing, but you did another, creating an individual with birth defects, shortened lifespan, deleterious consequences. Another type of off-target effect is that the change in DNA you make the right change, but it has consequences you did not envision. This might not be as big a concern when you're changing a deleterious allele to wild type. We know what wild type allele looks like. But it could certainly be a concern in introducing DNA from another species or synthetic DNA. Pleiotropy and epistasis. The changes that you make, though successful, have other unintended consequences due to the fact that genes don't operate in isolation and they don't operate in only one place. Plus sort of a range of things. These are safety concerns. These are sort of familiar stuff. And to a certain extent, we have things in place that look at this, and we're kind of good at dealing with them. Other types of concerns, what happens if we succeed? Well, we have to think about loss of diversity. So we talk about loss of diversity to the whole gene pool, which is definitely a problem, but that would be a huge level of effect, and it seems very unlikely to happen. Because even though you could picture it like vaccinations, it involves an IVF procedure, so it means that sort of the old-fashioned type of reproduction suddenly doesn't happen. And that sort of like giant effect across the entire human gene pool seems at least very far off to me. But changes to discrete populations to create more homogeneous groups, that is a much bigger concern to me sooner. George Church has a quote recently in New Scientist where he said, "I don't think everyone will want the same thing. We won't all suddenly become 5 foot 10 inches tall with blonde hair and blue eyes. I think augmentation will actually increase diversity." I would just like to say that apparently George Church did not attend my junior high school. I went to see the Steve Jobs movie recently, like last week, and I remember walking out of there and thinking, you know, it would really be a worse world if there were no Steve Jobs. And also, it would really be a terrible world if everybody was Steve Jobs. We need diversity. We need diversity of talent, and we need diversity of temperament. Another concern about the loss of diversity is the locking in of current trends, prejudices, and so on. I want you guys really quick to just close your eyes and picture an engineered baby. Okay, open them up again. How many of you were picturing a baby that wasn't white? How many of you were picturing a baby that wasn't straight? So then you maybe you get my concerns. There is a reason why surveys show that we are uncomfortable with selecting for traits. Another category, deepening of social divides. Perhaps worse than the loss of diversity is the loss of tolerance for diversity, as something that could happen to anyone turns into something that only happens to some people. And for those people, are we going to be as generous with resources? Are we going to be more stigmatizing? And I think we can even look at this at the fringes as something that can look at with Down syndrome today. Is Down syndrome moving into territory of something that happens to some people and not others? It's an issue. It's an upsetting issue. Socioeconomic advantages turned into biological advantages. Will we create a permanent biological underclass if this occurs and there isn't equal access? And, when this occurs, I'm betting there isn't equal access. So that's a concern to me. It's a very undemocratic thing. Could alterations, here's your far off, like, blah, blah, comment for the day. Could alterations to the germline eventually result in speciation? I don't know. Finally, misuse of the technology. So, will this technology be used by individuals or by states for eugenics, for weeding out of undesirables? Could individuals or states actually want to breed in desired characteristics? Could they want to increase aggression or eliminate post-traumatic stress disorder to create better warriors, better fighters? Could states want to breed for certain types of intelligence at the expense of others that were profitable for the state but maybe not as good for society? So that's a smattering of issues, and these issues for me are more concerning than the first, mostly because we don't have really any mechanism in place to answer them. We don't have really any mechanism in place to make decisions based on big societal concerns that may be counter to individual autonomous decision-making priorities. And of course, we all have the nagging anxiety of anything that involves great power and unintended consequences, and the worry, even the presumption, that there is something out there that we cannot foresee, and that's the most frightening thing of all. And so for all these reasons, in 1975, the world, or rather that little piece of the world that dabbled in DNA modification, agreed that tampering with the human germline should be out of bounds. Of course, it is easy to decide to refrain from doing things you cannot actually do. And through the first decade of the 21st century, DNA modification, despite improvements in the technology, was far too inefficient and error-prone to be considered as a part of human reproduction. The first challenge to the line-in-the-sand status of human genome modification came in form of mitochondrial transfer therapy, as Carl mentioned earlier. It's called that, although in fact it involves not the transfer of the mitochondria, but the transfer of either maternal nuclear DNA or embryo DNA into a donor egg with healthy mitochondria. Proof in principle was established in primate studies that resulted in live births of cute little monkeys, and subsequently in human embryos that were not allowed to develop past the blastocyst stage. There's been an extended debate in the last several years in Britain about whether or not to proceed with experiments in reproductive use. Some of the negatives are the familiar arguments about safety. Is there enough data? Do the data suggest a high probability of success? Are there possibly better and safer ways to prevent the birth of children with mitochondrial disease, et cetera? But many people suggest that even if it worked perfectly, this is not something we should be doing because it alters mitochondrial DNA, which is still DNA, and the changes are heritable. So it breaks the rule. Those of you who've been following the debate over mitochondrial therapy probably know it better by its clickhole name, three-parent baby, a phrase that pretty much sums up why many people are not comfortable with the idea. A child with three biological parents does feel like a step into a different universe. It raises philosophical questions, but is that really what we meant when we made the rule? It turns out that the number one thing on BuzzFeed's list of things we don't know about three-parent babies is that when we talk about, is that the phrase three-parent baby is a bit of a stretch. Nice use of the headline, BuzzFeed. When we talk about biological parentage, yes, we mean DNA, but perhaps not literal nucleotides, not molecules, but the DNA as a physical manifestation of something larger, that aspect of ourselves that is controlled by biology. And in this less literal sense, does mitochondrial DNA make the cut? Does it make you a parent? Is it really enough for that? The BBC recognized the thorniness of this issue and shrewdly punted back in February, going from three-parent IVF to three-person IVF or three-person babies. So now that takes care of that problem. For me, the take-home, I don't want to go there. For me, the take-home message is that applications cannot always be foreseen. No one was thinking about mitochondrial replacement when the guidelines against germline modification were developed. And trying to shoehorn the debate into those terms actually reminds me of the contortions that lawyers go through when they try to articulate the position that the framers of our Constitution would have about, like, net neutrality. There is always the problem with lines in the sand and slippery slopes. There will always be an unforeseen application that needs to be judged on its own merits. But for the strict constructionists of bioethics, the value of having a line in the sand and sticking to it is paramount. In February 2015, the British House of Lords joined the House of Commons in agreeing that mitochondrial transfer experiments could go ahead if they were approved by their Human Fertilization and Embryo Authority. Groups in the US are also very interested in pursuing these options for families with mitochondrial disease, and a committee of the Institute of Medicine has a report due out in 2016. The essence of the argument against mitochondrial therapy is the slippery slope. The practice, if safe, might be innocuous, but it opens the door. It is the gateway drug of human germline modification. And then came CRISPR. Just imagine the pot is going to be like with CRISPR. CRISPR is a game changer. Settle down. CRISPR is a game changer. It is literally disruptive technology. I believe it will change medicine. I believe historians recounting the early years of the 21st century will say, Uber? It seemed like a big ideal at the time. It was like CRISPR, but for taxis. If mitochondrial therapy is marijuana, then CRISPR is crack. Cheap, effective, easy to get your hands on. I mean, I'm told. And it is a big deal to regulate how to decide how we are going to use it and how we are going to regulate it. I'm sorry, it is a big deal to decide how we are going to use it and how we are going to regulate it. The fact of the matter is that CRISPR will allow us to contemplate when, where, and for what reason we should be editing DNA in human embryos. There are a number of entities studying this issue right now, including the National Academy of Sciences, which is holding a symposium in December and forming a committee to study it for a year. And genetic counselor Kelly Orman is leading a group organized by the American Society of Human Genetics, which includes the representatives of professional organizations from the US, Europe, the UK, and Canada, so including the NSGC, with the goal of producing a unified statement on human genetic modification. So that's very exciting. And NSGC has put together our own task force to produce our own position statement. And we're organizing a webinar on the topic in January with the dual goal of educating our membership on the issue and getting feedback on where genetic counselors stand on the clinical use of these new technologies and what role our organization should play in the debate over public policy. So if this subject interests you, please look for that and participate. The idea of regulating human germline modification is probably something with wide support if we can extrapolate from earlier polls that looked at concepts like designer babies and embryo experimentation, although apparently not in this room, but in outside. Wanting a practice controlled and having regulatory options are two different things. There are plenty of players in the we shouldn't do that, that's unnatural camp who would see putting the decision in the hands of the government as the worst possible option. But if not the government, who? As for what is allowed right now, it is literally all over the map. This 2014 figure from a paper by Araki Anishi demonstrates the range from the darker pink countries where it is not possible to do human genome modification under law, to areas like China and India where guidelines exist but are not legally binding. You can see the US is marked as restrictive, but in fact, what we have are rules that control what NIH can fund, not what can be done. If you want to know what clinical practice that is developed solely by private funding looks like, think about IVF. Where regulations exist, what they regulate also varies from country to country. Some countries like Germany have very strict laws restricting research on embryos at all. Others like Canada have laws that specifically address human germline modification. Australia and France, among others, don't restrict germline modification of embryos so long as there is no reproductive intent and the embryos are destroyed by a certain developmental stage, about 14 days. The international scope of this survey also brings home the point that regulation in one country or region will not dictate the future of this technology. So in some sense, the question in front of us may not be, what do you want to see happen, but what do you want to see happen here? And the corollaries to that, do we harm ourselves by stepping away from research unilaterally if it exists in other places? Do we have to consider the risks and consequences of reproductive tourism, including less accountability for safety, less ability to do follow-up, and an exacerbation of the issues around inequalities of access? The dividing line between reproductive the thing is that it's not a yes or no question. There are lots of lines you can draw. Should we stick with no germline modification? Can we permit it, as in the case of Dream Drive, where you're simply converting heterozygous alleles to homozygous alleles? So you're not changing the DNA that the person has, you're just making both of their alleles the same. Is it okay if we take a deleterious variant and swap it for a wild-type allele? That's more familiar. Is this different from swapping a normal allele for a preferred allele? And what about introducing an allele from another species? And finally, what about introducing synthetic DNA? Earlier, I talked about dividing up the issues surrounding DNA modification by horror movie, and I'm going to stretch the definition of a horror movie a little bit and adding one other set of concerns that accompanies the great success of the new CRISPR system, or now I should say systems, because there's a couple different ones. And those are what I call the X-Men issues, the potential negative effects of fear itself. I have a fear of the unknown. Many scientists advocating for a moratorium right now are concerned about public perception more than actual scientific work and the corrosive effects of nonspecific fears. So they warn that early mistakes could lead to shifts in public opinion, stigmas, which could undermine all the potential benefits of other types of genetic engineering, and that kind of generalized fear like we see with GMO foods. This alone may be a reason to make human germline modification off limits. In that same category of X-Man fears, I would put the danger of secrecy and isolation. The lack of public funding, which is the standard US government approach to anything controversial in medical research, may mean that there is less transparency and less accountability as private companies move forward with secretive and unpublished experiments. And regulation may be seen as preferable, but total bans will cede all future work to areas of the world where they don't have these restrictions, and perhaps leave us in a position of falling behind in our ability to understand, to use, or to detect this technology. Horror movies may help us to understand what animates our uneasiness with human germline modification, but in real life, identifying reasons to be fearful doesn't end the conversation. You can't just say, Gattaca, like it's the end of the discussion. There are very good reasons to be cautious. I also bet the first time somebody came home with fire, their grandfather said, "Hey kid, get rid of this stuff, it burns." The question is not, are there risks? There are always risks. The question is, are the risks worth the rewards? So I'm going to go back to well, it's almost useless because they were so all together. Well, we'll try it one more time with the polls. I totally I thought you would answer differently on the first polls. Can we go back to the Poll slide, great. Okay, poll number 1. Which forms of human germline modification do you believe should be permitted? Okay, so the very few, wait, no, okay. The people in A and B must be less certain of their answer. It's the only explanation. So no reproductive intent, has a little bit, none. I think it's similar. We've done nothing. Okay. So the next poll, can we move to the next poll? I don't want to take too much time on this. Which statement most closely reflects your view of mitochondrial replacement therapy? I think maybe a little bit higher on the it is German. No, it's pretty much the same. Okay. The data's the data. No change. How are we handling the questions? You have the Twitter questions? Microphone stands and they have Twitter questions? Well, I'm going to sit down and...
Okay, we have a couple questions here. What role do you see genetics counselors playing in the utilization of CRISPR moving forward?
Well, we're going to have to explain it. It depends on how it's used, right? But I think the first thing that's going to happen is that people are going to walk in saying, I read about this, right? You're going to get that. That's the very first thing. So bone up. You're going to have to tell them what it is and that we're not right there yet. That's step number 1. And I hope that genetic counselors, it's cliche to say that scientists shouldn't make these decisions for the public, and it's absolutely true. But scientists shouldn't step out of the debate either, because we do have some knowledge, and we're in a pretty interesting spot because we are both able to understand the technology, and we deal with the families, right? So the people that might get helped, we can maybe speak for them. So I say the role genetic counselors are going to play first is explaining it to the public and participating in the policy debates.
And I would add that in talking with people, it'll be important to make a distinction between the kinds of CRISPR news that people read about. So I focused in my talk mostly on CRISPR on embryos, but the fact is that when people read about the successes of CRISPR right now, what they're reading about is really somatic CRISPR. So, and there are a lot of promising applications of that. I mentioned the organ transplants, but cancer immunotherapy is one possibility. Take someone's immune cells out and use CRISPR to basically give them the kind of genetic wherewithal they need to attack tumors, put it back in, send them on their way. That's one application that's being developed right now. And then you can imagine other applications that could be used kind of like the way that gene therapy has been developed. So gene therapy is, where you use a virus to insert genes basically into the nucleus, maybe get them into the DNA. CRISPR could be sort of gene therapy on steroids. So people may say oh my gosh, they're doing all these things with CRISPR, so of course you should be able to help, us with this, pregnancy issue. You got to say well, you were reading about a different kind of research on CRISPR. So it would be good to draw that distinction.
Hi, my name is Brianna Roscoe. My question is, you talked a little bit about regulation and who should regulate and how should we regulate, but do you foresee that some of the patent battles that are currently going on from the people who have developed CRISPR, MIT, some of those academic organizations, do you foresee that we may be tied up in patent battles on the clinical aspects of how CRISPR is going to be used in the future? And is that an acceptable form of regulation?
I'll take it.
Okay.
It's a great question. There are some, neither of us talked on the battles that's going on for either the patent or the Nobel Prize, both of which are pretty heated. there's a new enzyme, there are more enzymes coming out. And what I foresee is actually, if there was only going to be the one CRISPR-Cas9 system, that license could be potentially very expensive and the patent battle could be very intense. But as they're developing alternative systems, I think that's not going to be a big problem. I think that'll be a big issue for the people involved, but not for the field.
Just to go back to a little bit of the history. So basically when Jennifer Doudna, Emmanuel Charpentier, and others figured out how it was going on in bacteria, there was a race going on to start working on this in mammal cells and human cells. And then there was filing of patents for that in particular. And now there's a dispute with Feng Zhang. Basically, it's Feng Zhang versus Doudna and Charpentier, and I believe that's still in the courts right now. But then, but that was just focused on this CRISPR system that came from one species of bacteria. There are lots of bacteria out there, and they've evolved lots of different ways of using CRISPR. And so Feng Zhang and some microbiologists said, let's go see what else there is out there while we're at it. Turns out there's some other bacteria that make like really tiny, really efficient forms of CRISPR that it might be better than the original ones. That was, I believe, last month. And then last week, they published like a whole suite of other CRISPR systems. It's just, this is a flood. But it's not really different in a sense than gene therapy, because gene therapy is based on viruses. And, the first generation of viruses were not very good. That's why there was a death in one of the trials that nearly brought gene therapy to a halt. And since then, they've gone out searching for lots of other viruses in nature, and they've found much better ones, which is why we're starting to see gene therapy finally move towards FDA approval.
I think the cost of therapeutics generally is going to be a huge issue for us, but not particularly this patent, I don't think.
Hello. I have a question about the line in the sand slide. So I think another possible line in the sand that I'd love to get your thoughts on is this question of what's governed by genes. So you could say that we understand what the 20 or so thousand genes in the genome do at a high quality level, right? What's high quality? You could say the answer to that is 0, or you could say maybe we understand 100 or 200 genes at a high quality level. We don't even know what the other ones do. If there were, we don't know if there are genes that are associated with telling a joke well. We don't know if there are genes that are associated with so many other traits, religiosity, who knows what. And so how long should we wait before we fundamentally understand that project, the whole gene, environment, nature, nurture part of it, before we even get into saying what we should be able to do with the technology?
Every gene you mention, I think. Would I edit that one? Yes. Yes. These are the same problems we're going through in every other way that the exploding technologies, right? That our ability to do and our ability to get information is outstripping our comfort level with what we really know. And I think that's a part of the moratorium question. It's not necessarily that people feel let's not do this, but let's not do it fast, because fast is going to get us into trouble. We don't know enough. We always think we know more than we do. I don't know.
I've actually I used to have that attitude, but I don't have it so much anymore, just in sort of a practical sense. It's in the sense yes, the genome is this wonderfully complex network of interacting parts, and it's going to take forever to figure it out if we ever do. That's true. But there are lots of cases where, sort of in a practical sense, you can learn enough about it to intervene. So, muscular dystrophy, it's there's one of these sort of single gene cases, but even with complex traits, I think we're going to find that we may not be able to totally decipher the whole thing, but we can find out enough about it to manipulate it in a very strong way. There are actually there's a new generation of potentially incredibly powerful cholesterol-lowering heart disease drugs that's going to be coming out soon. That came out of studies looking at genes associated with low cholesterol. There's this incredible number of them, and we don't really understand how they all interact, but somebody said, I like this one. I like this gene. Let's look at this, and let's see if we can figure out a target on this one. And they discovered a really powerful drug. So you could take that same logic to CRISPR and say, like, okay, yeah, heart disease is very complicated, but what if we tinkered with that gene? Could we give people a much lower chance of developing heart disease through life? And rather than having to pop a pill every day, you're just born that way. So, you know, these sort of, biology is complicated excuse I don't think it's an excuse anymore.
Sometimes it is an excuse.
That's just how I feel.
I just also think that for the safety issues, and that's fundamentally a safety issue, are you picking the right genes and stuff, we have some built-in checks on that. It's a giant generalization, but parents are fairly conservative about what they do with their children. I can't imagine that there'd be a giant rush to do things that are unproven and highly experimental.
What about these stem cell clinics?
Those are people in extremis, right? That's not making decisions for their children. And people are much more conservative for their children than they are for themselves. Witness, well, everything, right? Like, I certainly don't want my children to behave the way I did. But we understand something about safety issues. They're not entirely unfamiliar. We have systems in place to look at, are things safe enough to go forward? That's a thing we do all the time, and that's a decision that medical caregivers do all the time, and that's a decision that parents do all the time. I think the unfamiliar ones are harder to know what to do with.
Okay, I think this might be our last one. And I think this is one that all genetic counselors struggle with on different things, but could you argue that reducing or eliminating certain diseases could also attack our diversity? So what we talk about with Down syndrome or whatever disease it may be, is eliminating the disease actually affecting our diversity?
I'll let you do that one because I already said it.
Wait, so you do think it reduces diversity to eliminate diseases? I mean, we're talking about genetic disorders.
It depends what it could. So the thing is, you're saying disease, right? And that's the problem, is like, how are you going to define that?
Right. If people start calling Asperger's a disease, then
absolutely
we're good. Yeah, that's going to have a dramatic effect on human society.
Right. It's always hard in the details. It's always hard in the details. Everybody says, I'm good with ruling out disease, and I'm more uncomfortable with selecting for traits. But it's hard to tell the difference, the two of them, in the, when does intellectual disability stop being a trait and start being a disease, when you really come down to it? Pick a number, right? It's really hard, and it's sort of a sliding scale. In some families, it might look, one thing might really seem like a pathology. In another family, it might seem much more normal. So,
and also,
it's a terrible thing to say.
Yeah, I guess all these issues and a lot of things that you were bringing up are population level questions. A lot of the sort of discussion about ethics is like, well, even if one person does it, there's an ethical issue that's raised. But these questions about diversity of the species or a new species, those are population level issues. And then you have to ask, is this really going to be something that's going to be happening at a population level? Which you have to be saying, like, are people who make less than $1 a day going to be getting CRISPR, IVF, whatever treatments? I mean, I just I think that maybe these issues are really truly hypothetical, because until the world is in general affluent enough that these are really things that they could consider, then these population level questions to me seem moot. I don't know. What do you think?
No, I would sort of say the same thing. I think it's, within small groups, though, you could be looking at it like, if this population that uses the technology could separate itself from other populations that don't use the technology. But if you're looking at, are we going to affect the human genome, the human gene pool, I just can't imagine that happening anytime in the near future.
No.
So I want to thank Laura and Carl for a thought-provoking talk.